let's see now. So Friday, so there's going to be the Friday quiz at the end of today's lecture on cellular respiration and the parts of signal transduction that we covered on Wednesday. And then next week on Monday is exam number 3 for you guys. Alrighty. Is it 3? I think it's 3.
On Monday, right?
Monday, yeah.
Yeah, but we're not. So signal transduction.
Signal transduction will not be on exam number 3. Correct, it'll be on exam number 4. Let me do a brief recap of where we are in signal transduction. The basic idea is that sometimes cells need to communicate with each other. And let's say this is the cell that wants to send the signal, and this big half circle over here is going to be the cell that needs to receive the signal. The way cellular communication works is this. Well, I'd like Monterman to draw at the board. The cell that wants to send out a signal, that wants to communicate with the other cell, releases a molecule called a signal molecule. And that molecule will travel to the cell that's going to receive the signal. There's a little vocabulary drill here. If the signal molecule just travels by diffusion between nearby cells, it's called a what? Begins with a P. Paracrine, right. Just by simple diffusion between nearby cells, it's paracrine. But if it's carried in the bloodstream from one cell to another, it's a what? Begins with an H. Yeah, it's hormone. Yeah, hormones travel, or signal molecules that travel are carried by the bloodstream, and paracrines are signal molecules that just drift from one cell to another through the cellular, through the tissue fluid. Okay, so a signal molecule is released. The cell that's going to respond to the signal molecule has to have a receptor to detect that signal molecule. And once the signal molecule binds to the receptor, there are changes inside the target cell that eventually lead to the activation of what's called the cellular response enzyme. I'll just draw that. The cellular response enzyme is the enzyme that actually carries out the response that the cell is supposed to have. Each target cell has some pre-programmed response of what it's supposed to do when it binds that particular signal molecule. And I gave you an example, liver cells, when they bind the signal molecule epinephrine, their response is to start releasing glucose out of the cell. There was another example. There's a signal molecule called histamine, and when cells that make up the wall of your blood vessel bind histamine signal molecule, what those cells do is actually shrink themselves to a smaller size to help make the capillaries leaky in that area. Anyway, each cell has what's called a cellular output response, which is the response it's supposed to do to the signal molecule. And there's always one enzyme, or maybe just one or two enzymes, that carry out that response, and those are called the cellular response enzymes. But where things get interesting is this. How is it that we go from the receptor binding the signal molecule to activation of this cellular response enzyme? Well, as it turns out, there are a group of proteins called relay proteins that span the gap from the receptor to the cellular response enzyme. In other words, imagine relay protein number 1 right here. When the receptor binds the signal protein, relay protein number 1 gets activated. And then relay protein number 1 then goes over and activates relay protein number 2. Now relay protein number 2 is activated, and then relay protein number 2 then runs over and activates relay protein number 3. Which is now activated, and then that finally gets around to activating the cellular response enzyme, and the cellular output response occurs. All right, so these proteins here are called relay proteins. I guess they're like a relay race where you tag the next person. And the whole thing, all the proteins involved, Gesundheit, from the receptor through all the relay proteins down to and including the cellular response enzyme, all the proteins involved are part of what's called signal transduction, and that is what we're talking about in this particular chapter. When we say a protein gets activated along this pathway, what does that mean? Oftentimes it means that a phosphate group is attached to it. Not in all cases, but in many cases, an enzyme or a protein that's not in an active state becomes active if you attach a phosphate to it. And that can be called phosphorylating a protein, or there's a term kinasing a protein, which means the same thing, adding a phosphate to one or more of its amino acids. So, tell me that looks vaguely familiar from Wednesday. Okay, good. All right, so there's been a lot of research, cellular research, to analyze these signal transduction pathways. And what they found in researching them is that there are three main families of signal transduction pathways, which can be characterized by three different types of receptors that you can find for these signal molecules. And the three different varieties of receptors are listed up there. There's a type called a tyrosine kinase receptor, a type called a G protein linked receptor, and a type called an ion channel receptor. And we covered this one last time. Let's look at that, the TK receptors. And this will be our diagram. Here's a cell, there's the cell membrane. The top will be the outside of the cell, and the bottom will be the inside of the cell. A tyrosine kinase receptor would look something like this, like that. And they always have some tyrosine amino acids as part of their intracellular domain. Tyrosine is just one of the 20 amino acids. And the signal transduction begins like this. The tyrosine kinase receptor binds the signal molecule. Nice little binding site for it there. But then what has to happen is a second tyrosine kinase receptor, which is also bound the signal molecule, has to come along and click together with the first one. And that's called forming a dimer or dimerizing. And the reason why that's important is the intracellular domain of these tyrosine kinase receptors can have enzyme-like activity. It can carry out a chemical reaction just like a regular old enzyme can. But the active site is only formed when the two tyrosine kinase receptors click together. And so what I'm saying is, now that the dimer has formed, we're going to see an enzyme-like activity here in the intracellular region. And to be a little more specific, what that enzyme-like activity is it turns into a kinase. The intracellular domain is going to now has the ability to attach phosphate groups to amino acids. There we go. So kinases are enzymes that phosphorylate amino acids. And let's watch that in action for a second. The source of the phosphates is ATP molecule, which we know and love as our major energy source molecule inside cells. But here the ATP is not really acting as an energy source, it's just acting as a source for phosphate groups because it's got 3 phosphate groups per ATP molecule. Okay, and so notice that the dimerized tyrosine kinase is autophosphorylating, it's autokinasing itself in that it's attaching phosphates to its own tyrosine amino acids. I guess I should use the term that it is a tyrosine kinase, meaning that it prefers to add the phosphate groups to tyrosine amino acids. And what I want to point out is this is a bit unusual. Your cells have lots of tyrosine kinase enzymes. That's a fairly common type of enzyme, but almost all tyrosine kinase enzymes phosphorylate other proteins, not themselves. So bear in mind that this is a bit weird what you're seeing here. All right, well, the dimerized tyrosine kinase receptor continues doing that. It'll keep grabbing ATPs and getting phosphates off them until it has kinased all of its own tyrosines. Phosphorylate some tyrosine amino acids. Now, why is it doing this? Well, remember our goal here is to activate the first relay protein. We're still up here at this receptor, the tyrosine kinase receptor. Well, those phosphates that are stuck onto the receptor act as allosteric activators of relay protein number 1. Here comes relay protein number 1, and it touches those phosphates there, and that leads to the activation of relay protein number 1. And again, that's kind of weird because normally proteins are activated when they get a phosphate on their own amino acids, but for somehow this one's getting, relay protein number 1 gets activated when it just touches the phosphates on the receptor. Okay, so relay protein number 1 is now activated. It's going to run off and activate relay protein number 2, which activates relay protein number 3, which will eventually activate the cellular response enzyme. Now, I think I mentioned last time, don't get the idea that it's always exactly 3 relay proteins in every signal transduction pathway. I just do that for convenience. A lot of signal transduction pathways have even more than 3 relay proteins involved. Awesome, there it is. All right, so there's relay protein number 1, and it's now going to go down and activate relay protein number 2. Now, in what way did it activate relay protein number 2? Well, typically, it's phosphorylation again. Typically, relay protein number 1 is a kinase, and so it's going to add phosphate to relay protein number 2 to activate it. Phosphorylation of the next relay protein. Many of these relay proteins are tyrosine kinases. Let's have a close-up of that. Notice relay protein number 2 has a tyrosine amino acid on it. Here comes relay protein number 1, which we'll say is a tyrosine kinase. And so relay protein number 1 gets a phosphate from ATP, adds it to the tyrosine of relay protein number 2. So we would say this protein is a tyrosine kinase. And now relay protein number 2 is activated, and its job is now to activate relay protein number 3. How's it going to do that? Well, tyrosine kinases are one common type of kinase. There's another type of kinase called a serine-threonine kinase. Serine and threonine are also a couple of the 20 amino acids, and so these type of kinases are simply kinases that add phosphates to those 2 amino acids. And that's what we'll see next. There's relay protein number 3. Relay protein number 2, which is a serine-threonine kinase, comes up to it, grabs some phosphates from ATP, and attaches them to the serines and threonines of the next relay protein. Phosphate, and so now relay protein number 3 has been kinased, and away it goes to activate the cellular response enzyme. Now, just for clarity here, I'm not saying that relay protein number 1 is always a tyrosine kinase, and I'm not saying that relay protein number 2 is always a serine-threonine kinase. You can have tyrosine kinase, tyrosine kinase, tyrosine kinase, or serine-threonine kinase, serine-threonine kinase, or serine-threonine kinase, or tyrosine kinase, serine-threonine kinase, tyrosine kinase. Any sorts of combinations and permutations are possible. It really depends on which cell you're talking about and which particular cellular signal transduction pathway is being activated. And in some cases, it's not even kinasing that's responsible for the activation. There's also this concept of allosteric activators, which are small molecules that can activate a protein. In some cases, that's the method of activation. And that actually relates to this other blob of text right here, but I'm going to delay talking about this for just a moment. It's going to come up on the next type of receptor. All right, so let's watch the cartoon of it there. This is supposed to be a liver cell that I'm using for illustration. There's the signal transduction cascade. Sometimes they call a signal transduction cascade like this a phosphorylation cascade, because oftentimes that's what it is. It's one phosphorylating the next, which phosphorylates the next all the way down until you finally get to the cellular response protein, which in all likelihood is going to be activated by being phosphorylated also, like so. And then cellular response enzyme cuts up the glycogen in this case. Someone give me a question. You're all sitting there kind of silent today. It's a Friday. Okay. Well, let's go on then. The second type or family of receptors you see in these signal transduction pathways are called G protein